Hello, thank you for taking time to view my virtual presentation today. My name is Muralidhar Hedge. I am a professor and endo director of DNA repair in the Department of Neurosurgery at Houston Methodist. Today I am delighted to talk about the current research activities in our laboratory, particularly on the topic of DNA damage repair in brain health and disease, from mechanisms to innovative therapies. So let me start with a brief introduction about myself. I am a neuroscientist by training. I did my PhD in neuroscience in CFTRI, India, and as a visiting scientist in Max Planck Institute, Göttingen, Germany. And I followed it up with a postdoctoral training in classical DNA repair in University of Texas Medical Branch at Galveston, and a second stint as American Parkinson's Disease Association Fellow in UTMB as well. So this unique training in neuroscience as well as fundamental DNA repair helped me to start my own niche area of research when I started my own laboratory at Houston Methodist in 2013. And I started as an assistant professor and moved up the ladder as associate professor in 2016 and as professor and endo director in 2021. The goal of our laboratory is to advance scientific knowledge towards excellence in translational research in the area of human aging and brain diseases. Our primary emphasis is on delineating the molecular insights into the role of genome damage and its stability, as well as repair sequences or repair consequences in human brain disorders, and developing mechanism-based innovative treatment strategies for neuroprotection as well as neuroregeneration. So I am happy to tell you that this is a new era of DNA repair-based innovative therapeutics, particularly for brain diseases in the last couple of years. And I am happy to tell that our lab is not only participating in this, and we are leading these efforts both nationally and internationally. So we have an outstanding team of vibrant scientists, both senior and junior investigators. So we have Haibo Wang and Joy Mitra, who are junior faculty colleagues in my team. Manohar Kodavati, Velmarini, and Vikas are talented postdoctoral associates. We have MD PhD students who bring some clinical perspectives, like Vincent Provasek, who is a joint MD PhD student from Texas A&M and Houston Methodist. We always host undergrad and high school students like Leslie, and Pavana Dikshit is a senior research associate and lab manager in our laboratory. I should also mention that recognizing this emerging area of DNA repair as therapeutic potential in brain diseases, Houston Methodist created a new division of DNA repair research this year to support our laboratory activities. So this all started with our breakthrough publication few years ago, where we linked a new class of RNA DNA binding proteins which were implicated that time in neurodegenerative diseases, particularly motor neuron disease, to DNA repair. These were RNA binding proteins. We first time proposed that they bind to DNA and help in maintaining genome stability. And these are multifactorial diseases. Two important proteins which cover 95% of motor neuron disease, like amyotrophic lateral sclerosis, like TDP43 and FUS. We identified new role of these proteins in DNA repair. And our lab also focuses on Parkinson's disease and brain hemorrhage, again focusing on DNA repair in these diseases along with motor neuron disease. This is the first evidence from our laboratory came in 2018, where we showed this protein TDP43, which is a stress response factor which plays a very critical role in DNA repair, particularly in brain cells. So we found that as soon as there is a DNA damage, the protein jumps onto the site of DNA and repairs it. And if you knock down the protein or if you remove the protein, the cells accumulate huge amount of DNA damage. And we established this with cells isolated from human patients, mice models, and human tissue. And when you have a pathology or defect in this protein, the brain cells accumulate this DNA damage that leads to persistent or chronic inflammation and gradual neurodegeneration and age-associated neurological disorder. And this was published in a breakthrough paper and in the prestigious journal of PNS, Proceedings of National Academy of Sciences. We followed it up to show that TDP43 mutations in genetically inherited diseases, they cause neuronal dysfunction by reducing DNA break sealing. Like when a DNA is broken, the sealing activity is reduced. And in parallel, a related protein called FUS, which is mostly implicated in again familial or inherited diseases, plays a role in oxidative DNA breaks like single strand breaks, and we found that this combines with protein machinery and partners which are required to stabilize the DNA breaks and seal them. And you can see here in healthy brain cells, the protein jumps onto the DNA and repairs it quickly, but in diseased or mutant brain cells, the protein fails to repair that DNA. And here we went one step further. So we isolated mutant cells from human patients, like induced pluripotent cells, and we showed that that is a DNA repair defect, and then we went on to correct those mutations by gene editing technology and show that as like a magic, the DNA damage as well as repair defect disappear. And moving on to its consequences, now we identified several new mechanisms of DNA damage in neurodegenerative diseases. Next step is to take it to clinics, take it to the patients and develop innovative therapeutics. There are two challenges in this. One is having an appropriate model which mimics human diseases. That's one of the big challenges for brain diseases, because you cannot see what is going on in a live patient except some imaging studies. So one of our important challenges was to generate these models. In addition to patient-derived cells and mouse models, we also generated several new cell lines which can mimic age-associated brain disorders, and we also distributed this to all the researchers interested in these cell lines all over the world, and we licensed this on a non-profit basis to Sigma Aldrich so that it's available to all over the world. And the second challenge was to understand how our body or cells respond to this DNA damage and what is the consequences of them. So that's extremely important so that we can interfere and prevent these consequences. So I will give an example of our studies in DNA damage and stroke here. So we found that blood degradation products in stroke, they not only induce DNA damage, but they induce senescence. Senescence is like a dormant stage. So when there is a huge damage, cells go into a resting phase. And this was initially thought by several scientists that it's a pathology and you have to activate those cells. So but we discovered that this is like when a patient or person is sick, he needs to rest. And this resting is an intentional adaptation or protection by our cells. So the reason is when these blood degradation products are further cleared, it increases huge amount of free iron, a metal iron, and normal healthy functioning cells this cannot tolerate this iron. But if cells are resting, it will give a window of opportunity to store that iron in an inactive form. So just preventing that senescence will be more detrimental to the body. So in this study, we developed used a nanotechnology to generate a nanomaterial in collaboration with Rice and Texas A&M, and this is patented now. We developed an antioxidant nanomaterial which prevents this senescence, but we also added a metal iron chelator, so which prevents that iron mediated toxicity. So this is now ready to go into clinical trials. So as one of the implications of these studies, more recently Joy Mitra in our lab published that during SARS-CoV and COVID infection, there are tiny microbleeds and hemorrhages in the brain, and these blood degradation products again induce DNA damage and that dormancy in the cells. Many of the brain cells exposed to this virus undergo this resting phase, they reduce their function. This leads to kind of brain fog and other stuff. So this is an important study where we can use our previous knowledge in brain hemorrhage and use the same nanotechnology or that therapeutic proof of concept to reduce the COVID-19 associated long COVID and neurological disorders. So next is taking it to the patients. So the two challenges here is these are complex diseases, and we discovered that rather than when there is a DNA damage defect, rather than externally stimulating DNA repair, we found that because DNA damage is important, our body, our cells have developed multiple repair pathways. There are backup machineries under normal circumstances, jumps onto the DNA when the primary repair pathway is defective and repairs it. But in TDP-43 and motor neuron disease conditions, these backup pathways, although existing in the cells, they cannot be employed. The reason is this pathway switch factors which facilitate this transition are affected. So we found that rather than externally stimulating the pathway, so this is a roadblock for the cells to utilize the pathway although it's existing. So just facilitate the cells to overcome this roadblock. Basically you remove that pathway switch factor, then the existing machinery can repair the damage. So it's kind of a natural healing. So to understand how our body cells are responding and facilitate the cells to overcome some roadblocks rather than completely changing the repair pathways. And second is, there are these proteins are pathological and aggregates in the cytosol, and the way we know how these proteins aggregates and are toxic. The way you prevent this toxicity, those mechanisms are also required for some other pathways. For example, a perylation required to prevent this aggregation is required for DNA damage. So here we identified novel mechanism where we can specifically remove this toxicity in the site of toxicity, cytosol, without affecting these other essential pathways. This is important to prevent long-term toxicity of many treatment strategies, which is one of the main concerns in brain treatments. So this is the philosophy of our lab, based on the mechanisms. One is, developing innovative therapies based on actual mechanisms, understanding of mechanisms. Second is understand how our body responds to some, these stress conditions and facilitate natural healing, and using a site-specific toxicity targeted treatment to overcome these diseases. So just few highlights: we published more than 100 research papers, in the last few years, highlighting these discoveries. We also published, because this is an emerging area of study, we published several books so that it can reach students, families, and patient families and caregivers. And our research is supported by multiple grants from NIH and various other organizations, and of course from Department of Radiation, or Department of Neurosurgery and Houston Methodist. And we are also supported by donors and funders for some of the projects. And our lab is open to students and new generation of scientists interested to work in this area. We also request donors to support some of these projects so that this exciting area we can translate to clinics and make it available to the patients in a timely and efficient manner. So apart from the people in our laboratory who contributed to the studies, I want to thank our leadership in the department as well as in Houston Methodist for their support, and also collaborators in the United States and Europe, Asia, and all over the world who are experts in some of the areas of this research and who are participating in our studies. Thank you very much for listening to my presentation. I am Muralidhar in the Department of Neurosurgery and Houston Methodist Neurological Institute.